And thank you everybody for tuning in. This morning it's my real pleasure to introduce today's Cancer Center seminar speaker, Dr. Himisha Beltran. Dr. Beltran obtained her Bachelor's of Science degree from University of Oregon and her MD degree at New York Medical College. She did her internship and residency training at University of Pennsylvania, and then moved to Weill Cornell Medicine in New York for her medical oncology fellowship. And she stayed there and joined the faculty in 2011 as an assistant professor and rose through the ranks to associate professor. In 2018, she was recruited away from Weill Cornell Medicine and is now at Harvard Medical School in Boston as an associate professor. And she is also the Director of Translational Research in the Department of Medical Oncology at Dana-Farber Cancer Institute. So Dr. Beltran has really had a meteoric rise in her career, and I've been following her work very closely. She's best known for defining the molecular and clinical characteristics of a really interesting manifestation of prostate cancer that has really not received a lot of attention and is now a very active area of research, in no small part to Dr. Beltran's contributions in this area. And she's going to tell us about this neuroendocrine lineage plasticity phenotype of prostate cancer in her seminar today, and I'm really looking forward to hearing more about it. So Dr. Beltran, the floor is yours. Thank you very much for joining us this morning.
Thank you, Dr. Dem, for that very kind introduction. And it's really wonderful to be here, albeit virtually, and hopefully I'll get to meet many of you in person one day soon. So as was mentioned, my primary interest has been focused on studying treatment resistance in prostate cancer. Here are my disclosures. So for those of you that don't think about prostate cancer all the time, I think it's helpful to kind of step back and remember the scope of the problem. Prostate cancer is a very common cancer, the most common cancer in men in this country, and affects millions of men worldwide. And despite a lot of heterogeneity, particularly in early stages of the disease, metastatic prostate cancer remains a leading cause of cancer death. And there have been advances in treatment of metastatic prostate cancer, but we haven't really moved the needle as much as we could or should be doing. And I think there's really an opportunity to do even better. There have been exciting recent advances in metastatic prostate cancer. We now have new standards of care for metastatic hormone-naive prostate cancer with treatment and systemic intensification, radiation to the primary in some patients. There's been advances in genomics and molecular imaging, and PSMA PET imaging, for instance, was just FDA approved last week for prostate cancer. And the approval of PARP inhibitor therapy earlier this year, olaparib and rucaparib, for a biomarker-selected population of metastatic castration-resistant prostate cancer patients has really now made precision medicine a reality in prostate cancer. DNA repair aberrations at the germline or somatic level occur in a substantial proportion of patients, which is justifying tumor sequencing much more routinely. And so this is opening a number of new avenues and opportunities for precision medicine, an area I'm very interested in, which I won't talk about a lot today, but I think has been very exciting for the field. This is what the treatment landscape of metastatic prostate cancer looked like when I joined the field a little over 10 years ago or started working in this field. You can see androgen deprivation therapy really being the backbone of therapy, which it still is today. But there really was only one drug approved for castration-resistant prostate cancer, which was docetaxel chemotherapy. This is what the treatment landscape looks like now. Very exciting. We have many new drugs, different mechanisms of action, more choices for our patients, sometimes more challenges in figuring out how to sequence these drugs, but there has been a lot of progress. Prostate cancer is an androgen-driven disease. It's been many years, the early 1940s, when Huggins and Hodges first described this in seminal work. And I think that really for the first clinical trial of a targeted therapy, which is testosterone-lowering therapy, really established androgen deprivation therapy as a standard of care, as I mentioned, which persists today. It wasn't until the early 2000s where it was recognized that the androgen receptor is still active in castration-resistant prostate cancer, and so castration-resistant prostate cancers are not truly hormone refractory. This led to sort of an explosion of research on the androgen receptor, new drug development, abiraterone, which inhibits androgen synthesis, enzalutamide, and other drugs that more potently target the AR as an antagonist, and several other drugs in development, AR degraders, and other mechanisms to target this key driver in prostate cancer. So as we more potently inhibit the androgen receptor, many of these drugs started in later stages of the disease and have rapidly moved their way earlier. As you can imagine, the landscape of treatment resistance is also evolving. Most patients with prostate cancer progress with bone nodal metastases in the setting of a rising PSA. PSA is androgen regulated, suggesting some degree of persistent AR activation in their tumors. But we are starting to see more patients that have either primary resistant or rapidly refractory disease, unusual sites of metastases, low non-rising PSA, suggesting that their tumors may be less AR driven. This subset of non-AR-driven prostate cancers is really an area that I've been particularly focused on, which we think from autopsy studies and other metastatic biopsy studies is thought to represent about 15 to 20% of late-stage prostate cancers, and potentially increasing after treatment with these potent AR inhibitors. This is just a schematic just to show how resistance to AR therapies is not so dissimilar to resistance to other targeted drugs in cancer, ER, BRAF, EGFR. You can get secondary alterations in the target, and in prostate cancer, those are amplifications, mutations, bypass, crosstalk mechanisms, and then this subclass of target indifference, which we see not just in prostate, but also in these other cancers. And in prostate cancer, one extreme manifestation of this is histologic transformation from an adenocarcinoma to a small cell carcinoma. Here's an example of a patient where you have pre and post treatment biopsies where you can see histology looking a lot like small cell lung cancer. You start to see expression of neuroendocrine markers such as synaptophysin and low or loss of androgen receptor expression. This also happens in other cancers, most notably lung cancer. EGFR mutated lung cancers can transform to small cell carcinoma, small cell lung cancers at the time of resistance to EGFR targeted therapy with retention of the original EGFR mutation. So this concept that if we selectively pressure a key driver, plasticity and even histologic or a transformation seems to be a shared resistance mechanism. We often see loss of androgen receptor in these small cell carcinomas, these castration-resistant neuroendocrine tumors, and lower AR signaling. But you do see a spectrum of AR signaling even in these more typical adenocarcinomas. This is data from the University of Washington Rapid Autopsy Program, where just to highlight that when you look before 2012, before the introduction of these potent AR drugs, the incidence of AR negative prostate cancer in men that died from prostate cancer was about 10 to 15%. When you look afterwards, you can see a much higher incidence, including both neuroendocrine and non-neuroendocrine AR negative disease. So our working model of this process by which adenocarcinomas transform to small cells or AR independent disease continues to evolve, and there are there's a spectrum of this, and I think what we've learned through the years is this is a process, not an endpoint, and there may be an area in between where where androgen receptor is still expressed or the tumors have mixed features before they evolve to a AR independent or negative disease at the end. Here's an example of a mixed tumor where we see, this is a metastatic biopsy, where you see adenocarcinoma and small cell carcinoma kind of intermingled in the same tumor focus. When we stain this tumor for the androgen receptor, you can see in this case, AR is only expressed in the adenome. The small cell carcinoma component is AR negative. And when we start to look at early genomic events, such as the TMPRSS2 or gene fusion, which is a prostate cancer specific genomic rearrangement that we can detect in situ by FISH, we've always seen concordance between these two histologic elements in mixed tumors. And this was a study many years ago now, but I think this provided early evidence to me, along with some other studies in the field, that these tumors seem to have a clone arise from the same cell of origin, and potentially there is an evolution from one cell state to another. data from a metastatic biopsy program led by the West Coast Stand Up to Cancer PCF Dream Team. They did biopsies post-potent AR targeted therapy and identified 17% of their patients enrolled to have small cell carcinoma. But interestingly, in this study, you can see the androgen receptor expression was still there, and despite AR signaling being low. And so again, I really think it's a spectrum, and that perhaps loss of AR itself is a later event. So a lot of my research has been focused on understanding the biologic events that lead to this plasticity or this transformation to a neuroendocrine prostate cancer, developing therapeutics to treat this, or the plasticity or the disease state itself. But I think in order to be able to develop therapies, we have to really understand or develop ways to identify these patients, because I think this is still a big challenge in the clinic to identify patients that truly have non-AR driven disease. And you can imagine there are a number of implications for this, for patient selection for some of these newer AR drugs that are being developed, whose tumors are still AR driven, and selecting patients for alternative therapies, early intervention, or co-targeting approaches. So when I started studying this several years ago, I started to collect these tumors, and it was quite these small cell carcinomas, and I was quite surprised actually at the time that there was so much variability in pathology, in the tumor morphologies, even within neuroendocrine prostate cancer. And metastatic biopsies were not being done very often, especially back then, and pathologists were, I think, not really classifying metastatic tumors based on their morphology in a systematic or standardized way. So working with Mark Rubin, who's a pathologist who was at Weill Cornell at the time, we organized a pathology workshop that was in collaboration with the Prostate Cancer Foundation. We had pathologists from all over the country come, and I'm not a pathologist, but I was actually quite intrigued by how variable, even within expert pathologists, calling this disease state could be. And what resulted from this workshop was an attempt to try to standardize the nomenclature as a framework for future studies. And this nomenclature shown here on the right was incorporated into the WHO classification, which I think was a good step. But nonetheless, I still think there has been challenges, not based on necessarily just on the classification, but also on the disease itself. When we start, IHC was not in the classification. The classification is based on morphology. But when you start to add IHC, neuroendocrine markers or AR signaling downstream genes to try to better delineate these mixed tumors in particular, you see a lot of variability. You can see neuroendocrine marker expression in adenocarcinomas. You can see AR expression, as I showed, in small cell carcinomas. And so it has been quite a challenge, I think, for the field. Another challenge is that not all patients that you think are going to have neuroendocrine or small cell carcinoma have that. And here's an example of a patient who had rapid progression, low PSA, and had a biopsy that showed just adenocarcinoma, classical adenocarcinoma. And I think that there's been an attempt to try to term this as aggressive variant prostate cancer, which the MD Anderson group has done a really nice job in defining clinical features and trying to see if the molecular features of these tumors are more similar to the neuroendocrine features or that we see in the classical small cells. Here is a patient who, slides from a patient who died from metastatic neuroendocrine prostate cancer. You can see at time of autopsy, all the different sites have very similar morphology. We went back to his primary tumor, and he had a high-grade prostate cancer that he had had a prostatectomy years prior. And even on re-review, we found some areas of neuroendocrine differentiation. So potentially there are some high-risk individuals, even with the same cell of origin of these neuroendocrine tumors, that may have some high-risk elements even from diagnosis. So given all the challenges, I think, in the clinic with the histology and even the IHC or even the clinical features, I think there's been a big push, or at least from my side, to try to define the molecular features as an attempt to better delineate this subtype of prostate cancer. So in order to, again, recognizing that this is a process and a spectrum within advanced prostate cancer, the approach that we took was to really try to compare the extreme ends of the spectrum to, with the rationale that if we can identify the features that are most consistent in these pure neuroendocrine small cell carcinomas, we may be able to understand what happens in between. Early on, we compared, you can see the transcriptome of neuroendocrine prostate cancer with hormone-naive prostate cancer, and you can see we saw wide differences, genes involved in neuroendocrine lineage, as you expect, but EMT, stem cell pathways, a lot of changes compared to luminal localized hormone-naive prostate cancer. The next step was to really compare these neuroendocrine prostate cancers to castration-resistant adenocarcinomas that seem to be, strongly AR positive classical adenomas. This took some time to try to collect metastatic biopsies, collect serial samples, especially at a time where this wasn't commonly done for clinical purposes. But we established a cohort and performed whole exome sequencing, RNA sequencing, and DNA methylation analyses of, again, these two ends of the spectrum. And I guess maybe not surprising now, but at the time I was surprised that the genomic landscape of these two subtypes, histologic subtypes in prostate cancer were almost identical, despite being so different clinically and pathologically, and even by the gene expression profiling. Here you can see a copy number landscape with the axis representing chromosomes, and you can see gains and losses. And what we just did is we overlaid the adeno and the neuroendocrine patient exome sequences one on top of the other. And you can see with the areas of gray showing areas of overlap that there's nearly perfect concord overlap. We do see some areas of the genome preferentially altered in one or the other. AR gains here in adeno, and RB and P53 enriched in these neuroendocrine tumors, and this RB deletion and P53 either deletion or mutations. And this was notable because we know that RB and P53 are universally lost in small cell lung cancer. And it's been known for some time that the combination of RB and P53 loss can drive small cell carcinoma in GEM models, the TRAMP model, suggesting that these may be important for the pathogenesis of these neuroendocrine tumors in prostate cancer. In a triple knockout GEM model developed by Sheng Yu when he was with Lee Ellis and David Goodrich when he was a PhD student at Roswell Park. He's now a postdoc fellow in my lab. What they found is that knockout of these tumor suppressors results in a neuroendocrine phenotype, expression of synaptophysin, loss of AR, and molecular features that start to resemble our patient neuroendocrine tumors. But RB and P53 loss are very common in prostate cancer in general. This is data from the Stand Up to Cancer Dream Team, where you can see genomic aberrations in CRPC plotted against primary tumors, and you can see P53, P10, RB, some of the most commonly altered genes in prostate cancer, and not all of these. In fact, I think none of these in this particular study were neuroendocrine. So they are enriched in neuroendocrine, but they're not specific to neuroendocrine disease. And so likely there are some other cooperating factors that really drive the transition to a small cell or neuroendocrine phenotype. Interestingly, in lung adenocarcinomas that transform to small cell lung cancer, in this study, they looked at the adenos before transformation and found an enrichment in RB and P53 loss compared to adenos that don't transform, suggesting that in lung cancers as well, RB and P53 loss may be predisposing or facilitators of later small cell transformation. And this has not yet been fully established in prostate cancer. Here's an example of a patient where we had 3 different biopsies as he progressed from an adenocarcinoma to a neuroendocrine prostate cancer. In this case, we start to delineate the phylogenetic trees for these serial samples. And in this case, we do see early clonal alterations in RB and P53. And I think the exome sequences of these serial samples have provided more evidence of what we suspected early on, that these tumors evolved clonally from an adenocarcinoma precursor. As opposed to the very similar genomic alterations, the epigenomic landscape of neuroendocrine prostate cancer is quite distinct. This is DNA methylation profiling, looking at unselected methylation sites genome-wide. You can see that the neuroendocrine tumors kind of cluster together on an unsupervised analysis away from your castration-resistant adenocarcinomas. There were 3 adenocarcinomas that clustered with our neuroendocrine patients, and these were from patients that had low PSAs, rapid progression, suggesting that there may be clues in the epigenome, the DNA methylation profiles that identify AR independence, even independent of histology potentially. When integrating DNA methylation and transcriptome profiling, we suspect epigenetic dysregulation of genes involved in EMT, neuronal differentiation, developmental genes, things that we think to be important for this neuroendocrine phenotype. So overall, our data from that integrated analysis of these 2 different subtypes suggests an evolution of a neuroendocrine prostate cancer from an adenocarcinoma precursor, enrichment of RB and P53 loss, significant epigenetic reprogramming. And so it's very tempting to try to come up with a way to define these tumors by these molecular alterations. And we attempted to do this by integrating the DNA methylation and RNA profiling to develop a classifier that we applied to different data sets. You can see TCGA data set, for instance, which are untreated tumors having not showing elevated these NEPC scores. But when you start to look at these advanced data sets, this is an autopsy data set, that the castration-resistant prostate cancers start to trend towards lower AR signaling and neuroendocrine prostate cancer. On the right is a recent analysis from the International Stand Up to Cancer Dream Team, which is, again, a biopsy study of CRPC. And you can see that the neuroendocrine score identifies our neuroendocrine tumors, and most CRPCs have higher AR signaling, lower NEPC score. But you start to see these cases, again, in the middle that potentially are those in transition, and many of these had alterations in RB and P53. Another thing that we've been working on is to try to expand what we've learned in tissue biopsies to see if we can detect any of these features non-invasively, as we know that the biopsies are obviously quite invasive, and doing this serially in patients is not always feasible. So in work in collaboration with Francesca De Michelis, who's a computational biologist at the University of Trento, we've been studying cell-free DNA and metastatic biopsies of patients across the disease spectrum, and again, enriched for patients with this neuroendocrine phenotype. And we decided to take a comprehensive approach initially to do whole exome sequencing and whole genome bisulfite sequencing, recognizing that we would have to highly select our patients to be able to get sufficient DNA, cell-free DNA to do these analyses. But we may be able to start here as a way for discovery, as we really wanted to first figure out, are we able to detect in the circulation what we saw in the tissue biopsies? And if so, this could pave the way for a non-invasive biomarker to detect these neuroendocrine tumors. But we also wanted to use cell-free DNA as a tool to understand heterogeneity and to distinguish different mechanisms of clonal evolution. So what we found is that the adeno and the neuroendocrine cell-free DNA profiles were very similar to what we saw in patients. We see enrichment in tumor biopsies, we see enrichment in RB and P53 loss in our neuroendocrine, AR in our adeno. We saw generally high concordance between tissue biopsies and plasma when looking for these common prostate cancer aberrations, which is very similar to other ctDNA studies in the prostate cancer field. We've also been interested in differences and using whole exome allele-specific copy number analyses. You can start to plot out copy number A, allele A versus copy number allele B, to try to understand or identify subclonal lesions. And here on the top is an example where you see the biopsy tissue and the plasma sample at the same time point, looking nearly identical when you look at the sort of allele-specific copy number space. Whereas the second patient, you can see that there are a lot of identical things, but there are some alterations in the plasma that are not detected in the tumor biopsy. And with time, we start to see emergence of some of these subclonal alterations that I think may be important for treatment resistance in prostate cancer. We also compared the genomic profile of tissue samples and matched plasma and tried to calculate the degree of genomic similarity. And what we found is that when looking at gains, losses, and mutations, that the overall similarity of between plasma and tissue in neuroendocrine prostate cancer was higher than in adenocarcinoma. So if a plasma sample is thought to represent all the different metastases releasing DNA into the circulation, a higher similarity between a plasma and a single site biopsy suggests that there's likely less diversity or less intra-patient heterogeneity in neuroendocrine prostate cancer. We also did the same looking at tissue biopsies across patients and found that there was higher similarity in neuroendocrine prostate cancer compared to adenocarcinoma, independent of the biopsy site, suggesting that there's likely less inter-patient heterogeneity in neuroendocrine prostate cancer as well. Here's an example of two patients where we wanted to try to understand what are what changes occur with time. And we did this, we collected plasma at very short interval time points. Here, the first patient is a patient with metastatic CRPC, rising PSA of over 5,000, bone-only Mets, a clinical picture that suggests AR-driven disease. Second patient is a patient who's developing visceral metastases with a non-rising, relatively low PSA, suggesting AR-independent disease. And just to illustrate, the first patient, when we collected these sequential plasma samples, you can see that there are differences. And this, again, is looking at whole exome sequencing. You can see, for instance, the relative abundance of AR mutations changing with time, suggesting that there's likely multiple clones within the circulation with competing frequencies as this patient's progressing. But the second patient, you can see all the plasma samples look nearly identical, suggesting dominance of a clone that persists with time. And in fact, we had even gone back to his biopsy, his CRPC biopsy, before he had any clinical features of AR-independent disease, which also looked quite similar. So overall, I think our cell-free DNA analyses so far have provided some insights, early insights into how these neuroendocrine tumors are involved, of how they evolve. And so if I could put this into the context of what we already know in the field, I think generally the data in the field suggests that metastatic prostate cancer has a monoclonal origin, arises from a clone in the original potentially multifocal prostate cancer. These tumors acquire alterations with progression and treatment resistance, and polyclonal seeding further leads to intra-patient heterogeneity in the case of castration-resistant prostate cancer. What our data suggests, and we propose, is that at some point during this process, there's likely selection, clonal selection of a dominant clone in the case of neuroendocrine progression, and this homogeneity and heterogeneity is detectable through circulating tumor DNA analyses. This is still early data. We still need to build upon this, but I think liquid biopsies do have an opportunity in prostate cancer to help us understand this even better. I also mentioned that the neuroendocrine tumors are characterized by epigenomic alterations, and so we've been also doing evaluating cell-free DNA methylation using whole genome bisulfite sequencing. We see hypomethylated and hypermethylated regions in plasma, as we expected in tumors. We see high concordance between a methylation of tumor biopsies and cell-free DNA of plasma. When we do the same kind of unsupervised analysis with methylation sites, now adding the cell-free DNA methylation, we see also good segregation based on methylation profiles. And so we've been working to, here are just some examples of genes that are hypermethylated. Here you can see this gene SPDF that's hypermethylated in tissues and in plasma of neuroendocrine prostate cancer. This is a gene that's downregulated at the gene expression level. The neuroendocrine marker INSM1, hypomethylated and overexpressed in neuroendocrine prostate cancer. So we've been kind of building on our classifier, combining both genomic and methylation profiles now to try to develop a more targeted assay for cell-free DNA. We're very interested to understand how often and when these emerge. Are there patients that are developed this early? How does RB and P53 maybe facilitate or lead to occur either before or with DNA methylation changes? And when are emerging targets expressed, and then trying to use potentially in the future, think about using something like this for trials in this space. Speaking of trials, I think that there is an important unmet need to try to better treat these patients. They tend to be very aggressive, and there are no approved therapies for endocrine prostate cancer. We extrapolate a lot from small cell lung cancer. We give platinum chemotherapy for those patients that have small cell or these aggressive clinical features. But there is a need for a lot of labs actually working and early clinical development working at different approaches to try to target neuroendocrine prostate cancer. I'll just tell you about a couple of the targets that we've been working on. One driver of neuroendocrine prostate cancer that we've been interested in is NMYC, which can drive a neuroendocrine phenotype. We know NMYC as a transcription factor is challenging to target. So we've been developing approaches. We studied the role of aurora kinase in stabilizing NMYC and some of the available aurora kinase inhibitors for blocking this complex formation. Here you can see NMYC is upregulated in neuroendocrine prostate cancer, as well as a subset of CRPC, and not typically expressed in benign or localized disease. Working with David Rickman at Weill Cornell, he's developed a GEM model of NMYC gain and PTEN loss, which develops aggressive features, loses AR, starts to express neuroendocrine markers, and has histologic changes. And now more recently, and David has developed an NMYC RB loss mouse that is even more aggressive, develops visceral metastasis. And we've been using these models to try to understand the process as well as target some of the downstream events. DNA methylation, what David and his lab have done is they've looked at different foci within these mouse models and compared those to the patient data that we had generated. And we start to see emergence of these DNA methylation profiles, even within tumor foci of different histologies within these mice. So there's many different ways that have been developed to try to target NMYC. One that I mentioned we've been focused on is targeting aurora kinase A. Alisertib is a drug that was developed as a catalytic inhibitor of aurora kinase A, but also we found to disrupt this aurora kinase NMYC complex, targeting NMYC for degradation. And this has also been observed in neuroblastoma as well, an NMYC-driven cancer. More recently, a work by Bill Kalin and Matt Oser at Dana-Farber has shown, and also work by the Eli Lilly group has shown that there's another rationale to potentially target aurora kinase in neuroendocrine prostate cancer, and it's that they found that RB and aurora kinase are synthetically lethal. And this wasn't in prostate, these studies, but I think it also provides some, rationale for prostate cancer as well. So I led a phase 2 clinical trial of the aurora kinase A inhibitor alisertib for patients with neuroendocrine prostate cancer. The study was designed many years ago, so the inclusion criteria were quite broad. Not everybody had to have a neuroendocrine histology, as we at the time thought that if we would enrich for clinical features and do a metastatic biopsy, that perhaps we would capture more of this disease state. But there was a lot of variability in the patient inclusion. We did not meet our primary endpoint of 6-month PFS. But we did learn, I think, a lot from this study and identified some exceptional responders that we continue to study to this day. This is an example of a patient who had a PSA of 3, a neuroendocrine prostate cancer. You can see here with synaptophysin, aurora kinase, and MYC amplification, and he had stable disease on the drug for a little over 3.5 years. Here is a patient with de novo small cell carcinoma of the prostate, a very rare tumor, who had progressed on platinum chemotherapy, who had a complete response, including liver metastases. Here's another patient who had CRPC, who progressed with a PSA of 0.2, who had rapid decline in his CEA chromogranin and a complete response radiographically. And so these were, I think, encouraging readouts. And one thing that we've been excited about is that this patient here, we had developed a patient-derived organoid from his tumor before he went on therapy, and also a organoid from a patient on the trial who did not respond to the aurora kinase inhibitor. And these organoids were developed by Loredana Puka, who was a postdoc fellow in my laboratory at that time. The organoids had similar responses in the laboratory as the patients did in the clinic, and we've been using these organoid models for many things. Here you can see aurora kinase and MYC complex formation by a PLA assay. And normally this Millennium alisertib compound disrupts this aurora kinase and MYC complex, as I mentioned. When you look at the organoid of the non-responder patient, you can see very high levels of NMYC aurora kinase complex, and this is the drug that the patient received. And you can see that this was not disrupted using this drug, and there were various other compounds that we've been developing and trying to test using these models where you do actually see complex disruption. So maybe this could be a better readout preclinically to try to identify novel therapeutics. So these organoids add to our growing list of models in prostate cancer, which are not that many, but I think growing as far as in vivo models, and there's only one cell line available for neuroendocrine prostate cancer, so we've been using these again to test other drugs. We've characterized these organoids extensively at the genomic, transcriptomic, and methylation level. Loredana grew these as xenografts, and then we've used, again, to try to identify other targets and drug approaches. EZH2 is another target that we've been interested in, which is upregulated in neuroendocrine prostate cancer, as well as CRPC. We found that NMYC interacts with EZH2 and redirects its activity. I'm not gonna, so this is just, I'll keep going because I'm running out of time. But what we see encouraging data from the same triple knockout mice that was mentioned by David Goodrich and Lee Ellis, where if you treat these mice with that become AR negative with a EZH2 inhibitor, the tumors actually start to re-express the androgen receptor, and this resensitizes tumors to enzalutamide. We've also found in cell line models that there's modulation of some of these plasticity genes with EZH2 inhibition. In our organoid xenografts, we see variable responses to EZH2 monotherapy. Here you can see an in vivo study, despite on-target effect, we don't see dramatic responses. And so this was the rationale for exploring combination approaches. So we used these patient-derived organoids, both neuroendocrine and adeno, and I did a drug library screen of 129 compounds without or with a EZH2 inhibitor. We found in the single agent drug screening, things enriched in CRPC were things that we know are approved in prostate cancer: cabazitaxel, docetaxel, enzalutamide, and other hits as well. Platinum in our neuroendocrine, So that was, I think, some reassuring validation that these hits may be informative. And when we look at our combination screen, interestingly, we found the aurora kinase inhibitor alisertib coming up in our combination screen. And in fact, the same organoid that was not responding well to the alisertib had a better response, potentially with the combination of EZH2 inhibition and aurora kinase A inhibition, consistent potentially with the role that it's playing. Both of them are playing in driving NMIC. We've also been following the small cell lung cancer field pretty closely because there are a lot of similarities between this phenotype, both clinically, pathologically, and molecularly with small cell lung cancer. And you can see EZH2 and aurora kinase also coming up in small cell lung cancer. And DLL3 is another target that we had been studying. We found recently that DLL3, delta-like protein 3, which is a cell surface protein, is overexpressed in the vast majority of neuroendocrine prostate cancers, a subset of castration-resistant adenocarcinomas, and not expressed in benign tissues or localized prostate cancer. Again, this is a target that had been developed for small cell lung cancer with an antibody-drug conjugate and leading to a phase 3 trial in lung cancer. In prostate cancer, we find that we've developed a number of assays, but we find that they're rarely, DLL3 again is rarely expressed in these localized tumors, but when tumors do become neuroendocrine, you can see they really rapidly turn on this cell surface protein. We treated these tumors with the antibody drug conjugate, and we saw activity. And while this drug is not moving forward in prostate cancer, there are a number of other drugs that are being developed that target DLL3, both that are being developed for small cell lung cancer, but also for neuroendocrine prostate cancer, including a BiTE and a trispecific T-cell engager by Amgen and Harpoon that we've been very excited about. It also points to the role of Notch, and Notch is a cell surface protein, but it's involved in the inhibitory ligand in the Notch pathway. And we know Notch is an evolutionary conserved pathway that's involved in normal development. DLL3 is transcriptionally regulated by ASCL1. In neuronal development, Notch signaling is on in neural stem cells and turned off as tumors differentiate into neurons. And we also see that downregulation of the entire pathway in our neuroendocrine tumors, and you can see upregulation of ASCL1 in neuroendocrine prostate cancer. So Shangyu Q in my lab is working a lot on how we've often thought of Notch as an oncogene in prostate cancer, in early prostate cancer, but and he's been studying, how this could be potentially turned off in the case of neuroendocrine prostate cancer. Looking at those triple knockout mice models, you see, similar to patients, you see downregulation of Notch signaling, Notch2 and HES1, in these P10RB, P53 deficient models. When you look at even early stages where you see synaptophysin, a neuroendocrine marker expressed, this correlates almost perfectly with downregulation of these Notch downstream proteins. And he's been studying, could we potentially reactivate Notch, and what would that do to plasticity in the neuroendocrine phenotype? As a first step, he took these triple knockout mice as organoids, re-expressed Notch, and orthotopically injected them back into mice. This is the control where you see synaptophysin expression, low Notch, high ASCL1. And here you can see with re-expression of Notch, you see more expression of the androgen receptor, loss of synaptophysin, corresponding to lower Notch pathway, or reactivation of the Notch pathway. So I think that this is potentially involved in neuroendocrine plasticity, and there's a lot of ongoing experiments to try to really understand how is Notch turned off, what's the timing and cooperation of these events, how does this correlate with some of the other genomic and epigenomic alterations that I had already mentioned. In addition to neuronal development, we also see transcription factors involved in endocrine development, upregulated in neuroendocrine prostate cancer. INSM1 is an example of a neuroendocrine marker that's often used clinically that we see as overexpressed in neuroendocrine prostate cancer, and this is involved in normal development of the pancreas. What Shangyu has found is that when you knock out INSM1, you actually get downregulation of neuronal or neuroendocrine markers. But interestingly, you also get downregulation of some of these other key lineage transcription factors, ASCL1, NMYC, BRN2, and others. And so what we think is that these transcription factors that are all becoming dysregulated as these tumors go from an adeno to a neuroendocrine likely cooperate, and there's likely both neuronal differentiation and endocrine differentiation transcription factors that are leading to this end stream process that we see clinically. So our working model of lineage plasticity, again, continues to evolve, and we continue to try to build on this. And I spoke about some of the work in our lab, but I know that a lot of others are also working on this, and hopefully we're making progress in this space. So just to summarize, aggressive prostate cancer, aggressive subtypes of prostate cancer exist that are less responsive to AR pathway inhibition due to loss of tumor dependency on the AR. And the incidence of lineage plasticity in the neuroendocrine subtype seems to be increasing. These tumors share pathologic, clinical, and molecular features with small cell lung cancer, but they arise clonally from prostate adenocarcinoma during tumor progression. Our cell-free DNA studies show that we can detect some of these alterations in the circulation and identify patterns of evolution, and likely there may be early dominance and an emergence of a resistant clone through clonal selection. So I think transdifferentiation and clonal selection are not mutually exclusive. Our preclinical studies suggest that there may be a reversibility of this process, but there are so many open questions that still exist. What are the role of the epigenetic drivers, these transcription factors, the tumor microenvironment? How exactly is the androgen receptor lost? I think we still don't know that. And I think this represents a number of new opportunities for drug and biomarker development in prostate cancer. So I just want to acknowledge members of my lab. I mentioned Shangyu a lot, but there's others as well that are really highly focused on understanding this process, and many collaborators at Dana-Farber. I'll just highlight David Rickman and Francesca De Michaelis as I spoke about our collaborative work today, both with NMYC and the cell-free DNA analyses. So thank you. I'm happy to take questions.
Thanks so much. Just a reminder to folks, two ways to ask your questions. You can either type them into the Q&A module or raise your hand and then we'll unmute you. We do have some questions coming in already. If Dr. Dame, you'd like to take a crack at reading those off?
Yeah, it'd be my pleasure. I'll moderate the Q&A, and so please just continue to type into the box, and Nick can also help you if you decide to raise your hand. So Misha, that was a wonderful seminar. Thank you very much for sharing your published and unpublished work with us. The first question is, and this question popped up when you were presenting your ctDNA profiling. And the question is, what could be driving the clonal selection?
You know, I think it's likely just therapeutic pressure that is leading to that. I think you see, you know, again, I think some of these clones may even be pre-existing, and so although they evolved from adeno, you see Plasticity adenocarcinomas, but you know, I don't fully know, but I think it's probably due to therapeutic pressure.
Okay, I'm going to read a longer question. So there's a recent publication by Korn et al., which observed treatment of metastatic NEPC/CRPC with carboplatin and cabazitaxel was associated with better progression-free survival compared to treatment with cabazitaxel alone, only in patients who harbored tumor-associated genomic copy loss or loss of mutation of RB1, P53, and PTEN. Of course, this is the 3-gene target that Dr. Koo identified. Do you know if this is going to be the standard of treatment for the 3-gene mutated NEPC?
So I think it's a great question. I think we're thinking a lot about the role of platinum in prostate cancer, and we've, the Korn and colleagues at Aparicio at MD Anderson. In those studies, they defined patients for enrollment that had aggressive clinical features with or without neuroendocrine histology. And what they found is that there were frequently tumor suppressor losses similar to what we see in these neuroendocrine tumors. And as I mentioned, I think that they are neuroendocrine tumors. These tumor suppressors are not specific to losses, are not specific to neuroendocrine. I think we need more data. I think in prostate cancer, we tend to think about platinum for small cell carcinomas or for DNA repair, altered like BRCA mutated cancers. I think it's quite intriguing to try to use these as patient selection for platinum chemotherapy, given what the data that they found. Again, the inclusion criteria for those trials were not these, and I think another trial is being planned by that same group to try to use that. I know that, I don't know, Chuck Ryan is planning a study, or I don't know if he's on the call, but he, in hormone naive prostate cancer, of very aggressive therapies as well. And so whether these tumor suppressors, whether they're pre-existent or whether they can be even prevented with these aggressive chemotherapy approaches, I think is a really interesting avenue. I do think that platinum is kind of standard of care for these small cell carcinomas. And I think that the broader subgroup of patients is still yet to be determined.
Thank you. And I forgot to mention that question was posed by Sayek Gatak. And foreshadowing, the next question on the list is from Dr. Ryan. And his question is, do you think we should be doing serial cfDNA analyses in patients as they progress? And at what threshold should we say that they have NEPC based only on cfDNA and do things like add platinum?
That's a great question. I do think that the cell-free DNAs at the moment are not diagnostic, and whether I'm very supportive of doing sequential cell-free DNA, because I do think that we can learn a lot from that, whether it's in the context of a study or clinically. I think that I would, at the moment, not feel comfortable, I would probably use that as a trigger for a biopsy, rather than use that to treat someone within. So, some of the targets that I mentioned, like DLL3, for instance, that's involved in those that might not be expressed in patients that have RB and p53 loss without the neuroendocrine lineage. And I think it depends on the target that we're developing. If we're trying to if we're trying to sort of prevent plasticity, treat neuroendocrine lineage markers, and I think we still have a lot of work to do. But what I would love to see is more incorporation of these studies in all of our trials, not just neuroendocrine trials, because I think we're not going to be able to understand how often this is occurring and what the clinical impact is unless we do this in a larger scale, than even in cases that we don't have biopsies on. I didn't talk about imaging, PSMA imaging, FDG imaging, things like that. Even emerging newer targets for imaging can also potentially help. I think the field will benefit greatly from having a non-invasive way to identify different resistant subtypes beyond neuroendocrine.
And Chuck did follow up while you were talking to say that his study is underway with 13 patients treated so far. So I have a question. So you did demonstrate when you did your genomic profiling and comparing NEPC to CRPC that although AR gene amplification and AR gene alterations are less frequent in NEPC, they're not absent, they're still occurring.
Yeah.
So then you also showed that not all NEPC loses expression of AR. There is a subset that continues to express AR. Is there any sort of a correlation between those adenocarcinomas that amplified the AR gene, if they are able to transition to NEPC, if those are the tumors that retain some degree of AR expression, because it may be very difficult for the tumor to eliminate all AR expression from a gene that had been previously amplified?
Yeah, that's a good question. We don't have a lot of cases where we have serial samples to be able to systematically ask that. I know that the West Coast Dream team, for instance, they found a lot of AR expression and amplification in their small cells. And I think that, potentially if they had earlier time points could get at that a little bit more, but I don't know. I'm really intrigued by those cases that kind of persistently have those. and why they aren't, selected for. And I think there is some data to even suggest that AR potentially is doing something else in this context, being redirected and less dependent on the canonical pathway. So it is pretty interesting.
And then your comparisons, your DNA methylation profiling comparisons of NEPC to CRPC, you identified many differences. So I have two questions. What is the underlying mechanism? What's causing these changes in DNA methylation? And if you look at other epigenetic marks like histone methylation or those sorts of profiles, do you see any differences there as well?
Yeah, I don't know. We do see differences in all of them, the histone marks, as well as DNA methylation. We initially focused on the DNA methylation also because we could detect that easier using liquid biopsies. But we have expanded now to different, and they are providing a lot of clues into sort of activation and of marks and repressive marks on different pathways, but they are very different than adenocarcinomas. And I do think that's where, we need to expand a little bit more our knowledge into how they're being dysregulated and when. I don't know.
Wonderful. So I think that's it for questions. So please join me in thanking Dr. Beltran for a wonderful seminar today. I know I learned a lot and appreciated hearing your excellent work and look forward to seeing more. So thank you very much for taking the time to join us. And
thank you for inviting.
have a great holiday.
Thank you. You too. Thank you.